This lecture is over Chapter 13, Transcription and the Genetic Code. So today we're going to talk about what transcription is, the process of transcription. Then we're going to talk about post-transcriptional modifications that occur after transcription has occurred. And then we're going to talk about the genetic code and how the body uses the genetic code to code for amino acids. Just as a reminder, this is the central dogma of molecular biology. This is how the body goes from DNA to protein. DNA gets replicated and passed on and inherited to daughter cells and gametic cells, but then it'll be transcribed into messenger RNAs, rRNAs, or tRNAs that then get utilized in the ribosome to become translated into protein. Every living thing is made up of proteins. So proteins are the building blocks of life and cellular function. Transcription is where RNA is synthesized on a DNA template, and that genetic information that's stored in DNA gets transferred to the RNA, and it serves as an intermediate molecule between DNA and proteins. Now, we should note, out of the roughly 3 billion base pairs that we have in our genome, only about 1% is ever transcribed into RNA. So there's a lot of other aspects to our DNA that serve purposes besides becoming an actual protein itself. And a lot of those DNA motifs get utilized to regulate transcription. In order for us to engage in transcription, we need an RNA polymerase. This directs the RNA synthesis. We don't need a primer like during replication. And then we're going to need template, and that's where we'll start. And we're going to need a promoter site, that's our start site, and our terminator site. Now, we speak of transcription, a region that gets transcribed, we call it a transcription unit. That includes the gene and then the promoter and terminator sequence. The promoter sequence is where we start transcription. The terminator sequence is what tells the polymerase to stop transcription. In general, promoters are DNA sequences that are found around 30 to 40 base pairs away from the gene of interest. So in eukaryotes, that promoter sequence generally starts with what we call TATA. So it's a TAT triple A motif. We refer to it as the TATA box. And it specifies which strand is going to be used as the transcription template. Okay, so the TATA box is actually found on the non-template strand. So here we'll see, here's the TATA, right? And that means that we're going to utilize this strand for our template strand. Before we go too much further with transcription, let's back up and talk about RNA as a molecule. So ribonucleic acid is the difference between RNA and DNA is the fact that we have this extra oxygen on our second carbon in our ribose sugar, okay? The then nucleotide bases that we have that can attack, we have 3 are the same as DNA. So we have adenine, guanine, and cytosine. But then instead of thymine, we actually have uracil. And you can see the difference between thymine and uracil. They're very close. But the uracil has just a hydrogen coming off of this carbon, whereas the thymine has an additional carbon that's bound to 3 hydrogen ions. Okay, so uracil doesn't have that methyl group, and it can bond with cytosine, guanine, or adenine. Okay, primarily it's going to bind with adenine. Adenine can only bind with uracil, but it's also possible for uracil to bind with cytosine, guanine if it's needed. Okay, we then refer to our ribonucleoside triphosphates that'll get added during transcription as R-NTPs instead of D-NTPs because they're ribose, not deoxyribose. Okay, the phosphate group on RNA is going to be the exact same as in DNA, and in general, RNA molecules are almost always single-stranded. Okay, we're not going to make a double strand. It's just a single strand. Because they're single-stranded, they decompose very rapidly. Okay, you remember on DNA we talked about how the two anti-parallel strands create this bubble wall of water, in which you have water on the outside that protects the bases on the inside. Well, when you're a single strand, those bases, even though they're hydrophobic, aren't able to keep themselves protected as well, and so they're going to rapidly decompose. And as such, we use RNA as a messenger molecule because it has a short half-life. Any type of chemical interactions tend to break up that molecule. There's three main types of RNA found in the body. Okay, we have transfer RNAs, which we'll call tRNA. These serve the function of carrying amino acids to the ribosome during translation. We can have ribosomal RNAs, which we designate rRNA, and these actually are what form our ribosomes. So ribosomes are rRNAs, and they put amino acid chains together to form proteins. And finally, we can have messenger RNAs, or mRNA. These carry out the actions of a gene. These are the actual coded genes that will make proteins, are our mRNAs. As far as polymerases go, there's three kinds of polymerases. We have what are known as RNA polymerase 1. These are the polymerases that are used to transcribe our rRNA molecules. We have RNA polymerase 2, which is associated with our mRNA as well as RNA editing molecules. And we have RNA polymerase 3. This transcribes our tRNA. So make sure you know which type of RNA corresponds to which type of polymerase corresponds to which type of RNA. Now, the process of transcription begins with initiation. So initiation starts when an RNA polymerase, which is similar to a DNA polymerase, it's a holoenzyme, again, meaning there's multiple subunits. And one of those subunits is called sigma. So sigma finds the promoter region before the gene. So the sigma is going to bind to the TATA box. And it binds, and when it binds, it causes the RNA polymerase to clamp down and form a transcription bubble. Okay, so only the sigma subunit binds to the promoter. As the RNA polymerase then separates the double-stranded DNA molecule, we have a short section that becomes exposed in our transcription bubble, and at this point, the sigma subunit is going to leave the polymerase. Okay, and the polymerase then begins to add. So that's initiation. We then kind of enter into what we call elongation, in which we start adding our RNTPs. But in order for us to get from initiation to elongation, we have to form a small primer of sorts. It's not a primer in like how we talked with replication, but we need to get enough RNTPs added that this complex becomes stable. So oftentimes what we get is failed transcription, in which the transcription tries to start laying down a few RNTPs, but then it becomes dislodged and we get what we call abortive transcription, at which time the sigma subunit needs to reattach, reclamp down, create the transcription bubble again. But once we get about 8 to 10 RNTPs down, this is pretty stable, and elongation is going to be completed until we get to the terminator sequence. Okay, so finished initiation, that first RNTP keeps its 3 phosphate groups on there. All the other ones, those 2 phosphates will be cleaved off as we create the phosphodiester bonds. And in elongation, that's the sigma subunit has fully left, and we start adding RNTPs that match the template strand. Okay, unlike DNA, both strands aren't being transcribed. Just a single strand is being transcribed, the template strand. Okay, so elongation will continue as we add our RNTPs until the enzyme moves to the termination site. And that termination site is a sequence of DNA that's recognized by the recognized by the polymerase, and it causes the polymerase to stop adding RNTPs, and it detaches from the template itself. Okay, in prokaryotes, that termination usually causes the newly formed transcript to fold back on itself, and we create these hairpin structures that have hydrogen bonds, and that's what causes the polymerase to kind of stop. Okay, in eukaryotes, the transcription factor just directs the RNA itself to stop. So just to recap, let's go through the steps again. So transcription begins with template binding by RNA polymerase at a promoter. Okay, then the sigma subunit recognizes the Tata box, DNA double helix becomes unwound, we get a transcription bubble, and we start elongation. Okay, eukaryotes are promoter sequence Tata box. In prokaryotes, there's two promoter sequences. One is what we call we have what's called the Primnow box, and then there's this other sequence that has the T-GACA. Okay, and you'll notice that Primnow is pretty similar to our Tata box. Okay, and then elongation will continue until we reach the termination nucleotide sequence, and that causes it to stop. Our results, well, if we were transcribing a gene, then the result would be an mRNA. Okay, if the gene is a non-coding for a protein, it could be either a tRNA or an rRNA. After transcription, in eukaryotes, there's an additional processing that goes on. In prokaryotic cells, we actually take those mRNAs and they will be immediately translated. But in a eukaryotic site, we'll actually process these mRNAs a little bit further to allow them to last a little bit longer in the cell. So it also adds on some targeting sequences onto them. The 3 post-transcriptional modifications that I want you to be aware of in eukaryotes is the addition of the 5' 7-methylguanosine cap, the addition of the 3' poly A tail, and the excision of introns. So let's start with our 7-methylguanosine cap. So this gets added onto the 5' end. So if you remember, the first RNTP that we add still has its 3 phosphate groups that are on it. They were never cleaved off. And what we do is, in order for it to be, this mRNA to be recognized by the ribosome, we take that leading phosphate and we remove it. And we add to it we add the ribonucleotide guanine to that phosphate. Okay? And then we also add a methyl group onto that guanine and the first 2 mRNA nucleotides on that, and those methyl groups prevent decomposition from our nuclease. These are enzymes that kind of break down RNA. So we're adding stability, and we're giving it a recognition site that the ribosomes in the cell will recognize and say, oh, that is an mRNA, let's bind to that. Okay, this capping actually occurs usually before transcription is even completed. Okay, the second modification is adding what we call a poly A tail onto the 3' end. So the poly A tail is where we stick a string of adenines onto the end of our mRNA, and typically it's somewhere between 10 and 35 ribobases, and it's this very highly conserved sequence in which it's adenine, adenine, uracil, followed by 3 additional adenines. And it just repeats over and over again. Okay, why the cap and the tail? Well, again, the cap is used as a signal molecule, but RNA is also easily degraded. It's a temporary messenger. And by putting on the cap and the tail, we prevent the ends from being cleaved by exonucleases, right? So these nucleases, they break down those phosphodiester bonds because we're single-stranded. And so we can protect the ends with the cap and the poly A tail. They can also be used as signal molecules to have mRNAs degraded by very specific nucleases. So there's some nucleases that actually recognize this site and cleave it off, and that causes the mRNA to decompose really quickly. So the cell can target the destruction of mRNAs if it needs to, using the poly A tail. The last post-transcriptional modification I want to talk about is editing, or what we call splicing. So in eukaryotic cells, we have regions of DNA, right, that contain what we call exons. So I'm going to just put an E here. And in between, we have these intervening sequences that are called introns. The exons are the coding region. So maybe this is exon 2, this is exon 1, okay, intron 1, maybe we have another intron, so intron 2. And the intron, they're non-coding, so these do not contain sequences that should be translated into amino acids. There may be some regulatory DNA motifs in here that help. We'll talk later about, when we get to gene expression, about what these sites can do. But we need to remove these out because they're not coding. So when we splice, we remove out the introns and we just string the exons, or the coding regions, together. And this gets mediated by what we call spliceosomes. These are small nuclear RNAs, and they kind of help. Sometimes we call them SNRPs. They help remove out the introns. And what we find is that on the beginning of all introns, we have these sequences. They start generally with a GU, and they end with an AG. So these are called donors and acceptors. So again, we would have a GU site on this end and an AG over here. Two types of splicing. Okay, the first is kind of what we call a ribozyme. This is a self-splicing molecule. It's usually very small. They're classified as group 1 introns. So this intron is a short intron, and the RNA is capable of cutting itself off all by itself. So here's an active site. It binds a guanosine onto there, and then that guanosine basically is attracted to the donor site and the acceptor site, and it pulls itself out, all on its own. Okay, group 2 introns now are a lot longer. These are the ones that have the GUs, so our donor sites and our acceptor sites. And then somewhere in between, there's what we call a branch point, and we need multiple spliceosomes to be involved. So we have all of these SNRPs that bind at the donor site, the branch point, and this causes these to kind of be attracted to each other, creates loops, and gets excised out. Okay, so what I want you to know for spliceosomes, the difference between group 1 and group 2. So the difference between a ribozyme and a spliceosome. I want you to know what the donor sequence is and the acceptor sequence. So donor is GU, acceptor is AG. So this is what it looks like, right? We finish, here's our initial DNA. Here's the gene. We have a promoter region. Granted, the promoter would be on the opposite strand, but we're just kind of showing, right? And we would then finish our mRNA. So as we start to go, here's our 5'. This is the pre-mRNA. We then add our 7-methylguanosine cap, our poly A tail, and then we splice out all of the introns and just put the exons together, and this would be our finished mRNA. Okay, exon stands for expressed sequence, intron stands for intravening sequence. In some genes, we have what's called alternative splicing, in which we splice out introns and exons. And the benefit of this in these genes that have this is that we can increase the possibility of genes. Right? We can have a single gene, but if we cut out different exons, then we can have different amino acid sequences. We see this with a lot of our immune genes, okay, like an antibody. Okay, so again, this would be a traditional mRNA in which all exons are conserved. But here, if we got rid of 2, we have a different protein product, or here, a different protein product. So just to recap, again, on our molecular dogma, we're talking about transcription and how we get to the mRNAs. Now, the last part of this chapter talks about what we call the genetic code. So what the function of our nucleotide bases serve. Those nucleotide bases are a code for our phenotype. So the genotype is the actual letters. The phenotype is what gets expressed, what we see, and that phenotype is made up of our proteins, right? That's however we create those proteins, that's going to be, and the combination of those proteins is going to yield the expressed trait. Okay, so amino acids, those are the building blocks of proteins, and they create polypeptide chains. Okay, polypeptides can be made up of from just a handful of amino acids to maybe as many as 1,000 different amino acids. But there's only 20 different amino acids. Now the genetic code is written in a linear form, and it uses ribonucleotide bases that comprise our mRNAs as the letters. Okay, so here are the characteristics of the genetic code. First off, it is triplet. Okay, what it means is the words that we read, we have three ribonucleotides for every one amino acid. Okay, we call these a codon. So a codon is 3 bases. Okay, it is also unambiguous, and what we mean is that for every one codon we have, there is only one amino acid that it codes for. Okay, so you can't have a codon code for 2 amino acids. That being said, our code is also degenerate. That means, of our 20 amino acids, 18 of them are specified by 2 or more codons. I'll show you that just in a second, the difference between degenerate and unambiguous. Additionally, our genetic code contains a start and a stop codon. So the start codon is what is specific code, and it tells this is where you start adding on amino acids. The stop codon is a nonsense codon where there are no amino acids that code for it, and so nothing gets added and we stop translation. Finally, the genetic code is only read in one direction. Okay, we always read 5' to 3'. And it's nearly universal, meaning that code really doesn't change much between organisms. All organisms' codons are pretty much the same. There's some minor differences, but not that many. Okay, so again, let's look at, here's our amino acids. Again, we said it's read in triplet, so you'll notice that all of our codons have 3 nucleotide bases. Now, I said that the code is unambiguous, meaning each codon codes for exactly one amino acid. And you'll see that. You don't see UUA, uracil only makes a phenylalanine. It doesn't make anything else. That you'll find triple U nowhere else in this code. But we also said that the code is degenerate. And what we mean by that is, like, if you look at leucine, right, all of these codons will result in a leucine amino acid being added onto our polypeptide chain. Okay, our stop codon, the universal stop codon is AUG. It codes for the amino acid methionine. The three stop codons that we have are UAA, UAG, and UGA. These are all nonsense. You'll notice there's no amino acids that they code for. Now, you may ask yourself, how is the code degenerate? If you notice, for most of these, like let's take our tyrosine, or sorry, not tyrosine, threonine. Okay, threonine right here. You see how it all starts with AC, but it's that third codon, right? The third base that is degenerate. So you could have, it doesn't matter if it's a uracil, a cytosine, an adenine, or guanine, it's still going to result in this. That third position in the codon, the reason why we can get away with that is something called the wobble hypothesis, and it's due to the interaction between the mRNA and the tRNA. So on the tRNA, in that third position, it has an inosine, which means an inosine can kind of interact with a lot of different ones. And this will make a lot more sense in our next lecture as I talk about translation. Okay, so I'll come back and talk more about wobble hypothesis then. Now, each codon gets read separately, so they don't overlap. So what that means is if I start with here, right, if this is the initial frame, I'm going to read 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. I don't have overlapping in that. It's never overlapping. It's read continuous. You can think about these three letters as words, and there's no, you don't have any commas in there. We refer to that as the reading frame, and that reading frame is going to be defined by where that start codon is. Where the start codon is. So it could be these three, but if right here, let's just say this was an A, U, G, then that would dictate this is the start of my reading frame. So I'd go these three, these three, these three, these three, and so on. Okay, now we can have a couple types of mutations. We can have what's called an in-frame mutation. Now, in-frame mutation is where we only have either one amino acid inserted or deleted. So we can see right here, if we added this CAG in here, it doesn't necessarily change our reading frame. It's just adding in 3 base pairs, and so we would stay the same. But we can also have what's called frame shift mutations, in which we shift the reading frame. So if we stick in, so like right here, this is our frame. If we stick in a CAG into this position, and our reading frame is right here, it's going to shift everything. So we can see it right here. If we start in reading frame, but now we're out of reading frame. So GAC, AGG, that changes the amino acid sequence, and it changes all amino acids after that, right? So we're going to get a very different strand. That's a frame shift mutation. Okay, sometimes we can go in chain and then back into frame. That's possible that it can happen. I mentioned that genetic code is nearly universal. There are slightly different codes for mitochondrial DNA, plants, and prokaryotes. Those are kind of where we see our difference. There's a difference between plants and animals, prokaryotes and eukaryotes, and difference between nuclear DNA and mitochondrial DNA.